Good evening, everyone. Can you hear me? Do you want to hear me? Pretend I didn't hear that. Good evening. Welcome to the Los Alamos Historical Society Lecture Series. The series title is History Frontiers. And tonight we're in for a treat with a lecture on biosecurity. So given the topic, let me redo my greeting. Good evening, ladies and germs. My name is Todd Urbanch. I'm a board member and program chair for the Historical Society. I want to thank Los Alamos National Bank. They've been a sponsor of the entire lecture series. I asked a mutual friend if she knew anything about our speaker's work. She said no, she didn't, but our speaker throws the most phenomenal kids' birthday parties that you could ever imagine. Tonight's speaker is the Deputy Group Leader of the Biosecurity and Public Health Group in the Biosciences Division at Alamos. She received her BA from Columbia University, New York City, in biochemistry in 1991. She then went to graduate school at MIT and got her PhD in cell and molecular biology in 1997. She spent 3 years at Cornell University as a postdoctoral fellow before coming to Los Alamos in 2000. At Los Alamos, she has worked on a variety of projects in pathogen virulence and host response and immunity. Everyone, let's give a warm welcome to Dr. Elizabeth Hong-Geller.
So, good evening. Thank you, everybody, for coming. I'd like to thank Todd for inviting me and the rest of the Los Alamos Historical Society for me to give a lecture on the history of bioscience and also talk a little bit about the work that I do at the lab currently. So I came to Los Alamos in 2000, right after the Cerro Grande fire, and in the last 13 years, I'm continually learning about the rich history and breadth of bioscience here at LANL. So what I'd like to do tonight is to hit some of the highlights of the historical events of bioscience history, as well as talk about my own interests in host pathogen biology, and specifically in Yersinia pestis, which is the causative agent of plague. So Los Alamos is known the world over for the science and engineering of the atomic bomb. So major contributions in physics, in theory, in modeling, nuclear engineering. So I'm not stretching it too far by saying that Los Alamos isn't as well known in biological research, at least in the general public. What I wanted to show you is actually a trailer of this movie, Twins. It was made in 1988. Perhaps some of you remember it. It starred Arnold Schwarzenegger and Danny DeVito. So let me see if I can get this to play.
In a secret lab, somewhere in the Western Hemisphere, a perfect human specimen has been born. He has the strength of 10 men and the wisdom of 20. He also has a twin brother.
I have a brother?
Oh my goodness, this is good.
Arnold Schwarzenegger.
Born to be bad.
And Danny DeVito
Way to go, Mom.
are twins.
My name is Julius. I'm your twin brother.
Obviously. The moment I sat down, I thought I was looking into a mirror.
Only their mother can tell them apart. Twins, the new comedy from Ivan Reitman.
Julius, what?
Okay.
Something.
So, as you gathered, the movie is about Arnold Schwarzenegger and Danny DeVito's characters. They're twins, and they were born in a secret lab in the Western Hemisphere. That is off. Yeah, it's off. So they were born in a secret lab in the Western Hemisphere. And later in the movie, it's revealed that this lab is, in fact, Los Alamos. And as you probably recognize from some of the scenes, the filming was actually done here in Los Alamos in front of a DOE building that has since been destroyed, and also in Santa Fe as well. And the idea behind the movie is that Los Alamos is responsible for trying to create this perfect human being, and Arnold Schwarzenegger being the perfect human being, and Danny DeVito, the unexpected twin that was born. The movie has a happy ending. The brothers, disparate as they are, become buddies, and they get married to sisters in the end, and they find their long-lost mom in Santa Fe. But it certainly gives the impression to the general public, the perception of Lanel bioscience research as having to do with human cloning, the sort of genetic engineering. This is all fiction, we know, but this is what the general public is left with. And what I want to talk about today is the actual contributions that bioscience research at Lanel has made, and there have been a lot of them. So this is a timeline. It's not, I won't go through all the work that's been done here, but I do want to emphasize that the history of bioscience research stretched back into the 1940s with the developments of the atomic bomb. And in parallel, what we wanted to do was to understand the health effects of radiation. At the time, dose limits of plutonium exposure were not well understood. And so this work began in the 1940s. And there's been a lot of work since then. And to simplify things, I've made a more simplified timeline of what I want to talk about today. So I will speak about the health effects of radiation. This stretched for decades with a series of animal studies from the 1940s through to the 1960s. We also made great strides in development of instrumentation for biological research. So flow cytometry was invented and greatly modified here at Lanel. In the 1980s, the advanced computation that we had was started to be directed towards DNA sequencing and analysis of sequences. We started a database that later turned into GenBank, which scientists use every day in order to look at different DNA sequences from thousands of different organisms. We were involved in one of the original labs in the Human Genome Project to sequence the human genome. We were responsible for mapping of chromosome 16. With the turn of the century, we had some major events that happened in this country, primarily the 9/11 attacks and the anthrax attacks. So then we started talking about using terminology such as biowatch, biosurveillance, looking at mechanisms of pathogen interactions with the host and trying to develop new targets for drug development. And then later on, more recently, we've focused on other problems, therapeutics. So we've developed a novel HIV mosaic vaccine that has received a lot of press in the last year. And another one of our most recent focus areas is in biofuels. How do we develop new biofuels and not be as dependent on fossil fuels? So let's start with the health protection for plutonium workers. So back in the 1940s, scientists knew that radiation was going to be dangerous to scientists who were working with it. Back in the 1920s, a series of factory workers received radiation poisoning because they were painting glow-in-the-dark radium dials on watches. And so from that experience, there was a great deal of study in occupational health and safety in order to keep our workers safe. The early work focused on developing chemical methods to detect plutonium in human waste and lungs. We also did a number of animal studies to check detection methods, and then also tracer experiments on humans to determine the percentage of plutonium excreted daily. So these tracer experiments was a multi-institutional effort. It involved many different labs across the country. Eventually, 18 patients were enrolled in these human plutonium injection experiments. The human patients were located in other places, not Los Alamos. So in Berkeley, in Rochester, and in Chicago, these 18 patients were injected with tracer amounts of the plutonium. And then Los Alamos was more responsible for the analysis of the data that came out of these other laboratories. So these types of studies led to a reduction in tolerance limit from 5 micrograms to 1 microgram of total body burden of plutonium. We also found out from these types of studies that there was a different distribution of plutonium in humans compared to animals. Most of the plutonium ends up in the skeleton, in the bone, but in the human, we saw more retention in the liver. And from these types of studies, along with looking at other folks who have been exposed, there seemed to be a possible increase in osteogenic sarcoma. So this is a bone type of cancer. But most exposed workers seem to live longer than average. Other results from the radiation types of experiments done here at the lab was this Los Alamos Human Tissue Analysis Program. So this was started with the death of a worker, Cecil Kelly, who was exposed to a lethal dose of gamma and neutron radiation in December of 1958. And he died about 35 hours after that. But because he had been a plutonium worker for decades before, he had already a burden in his body. And so it was instituted that with the permission of the workers, there would be a collection of these types of samples from long-term exposures of the plutonium workers. So by the 1970s, there were over 100 samples from people who worked with plutonium, and they were combined with other registries to form this United States Transuranic Registry operated by the DOE, including hundreds of direct and indirect exposed workers. And the purpose of this program was to determine the body burden and retention of plutonium in the skeleton, the lungs, the liver, for modeling and prediction of exposures. So here is an autoradiogram of a bronchial lymph node from a former worker. And here you see these alpha tracks radiating in this star pattern from an alpha emitting material, most likely plutonium. So one of the more famous samples that we looked at was this case of Karen Silkwood, plutonium exposure. So this was made into a movie starring Meryl Streep back in 1983. Karen Silkwood was a chemical technician at the Kerr-McGee plant in Oklahoma. She had received a plutonium exposure, presumably an occupational exposure, in 1974. She was sent here along with her 2 roommates to Los Alamos for whole body detection as well as lung detection using this in vivo measurement lab at LANL, which still exists today in our HRL Bioscience building. It's one of these rooms where you lie very still for half an hour or more, and they do body counts. And what was found was that she was indeed contaminated with plutonium, whereas her roommates had very little contamination. She returned to Oklahoma November 13th, I believe, and then the next day she was killed in a fatal car crash. And so there's always been speculation that her death might not have been accidental. She was also very heavily involved in an activist in worker safety. And it was reported that she was on her way to deliver incriminating documents against her company the night that she was in this car crash. But be that as it may, her at the behest of the Atomic Energy Commission, some of her samples were sent to Los Alamos as part of this program to see whether it was in fact contaminated radiation. And the results were consistent with her in vivo measurement scans at LANL. So she did receive exposure. Apparently there was some radiation found in her gastrointestinal tract, so there was some ingestion of the material. In addition, what they were able to find was that some of this radiation was found in the inner lining of the lungs rather than inside the lungs. So that suggested that the contamination was relatively recent. So this type of work really contributed to the ability for LANL to detect radiation and also provide for hard data for worker safety. And Karen Silkwood has since become the face of this kind of occupational health and safety. So there were lots of different types of animal experiments that were done at LANL. One of the experiments was what was called these Astro Monks. So in addition to LANL interest in exposure of workers to plutonium, we were also interested in seeing whether travel in outer space caused some exposure. So back then, this was the mid-1960s. The Apollo 11 mission was still a couple of years away. And the idea behind this experiment was to train a series of monkeys, these Astro Monks. So there was a training program for monkeys in Holloman Air Force Base in southern New Mexico. These monkeys were trained to press a series of levers and buttons in response to a flashing light. So this is to simulate what an astronaut might have to do, their tasks that they might have to do when they're in a space capsule. So these monkeys were strapped in a harness, such as what you would find in a high-performance airplane. They were arrayed here in these little cubicles, and then a radiation source would raise from the floor, and the exposure would occur. So over a series of 10 days or so, the monkeys they would have some breaks, but there would be radiation exposure in order to see what the effects were. So if the monkeys were no longer able to press these levers according to what they were supposed to do, then that would be a sign that perhaps their faculties, their mental faculties were waning. So that was the experiment. This experiment was actually done twice. What happened was that in the first experiment, they used a particular species of monkey, this macaca monkey, because they were thought to be more docile and more easily trained. But what turned out to be the case was these macaca monkeys actually were padded more in their posterior. And part of the training program was that they'd receive a shock through their bottoms. So these monkeys were very smart. They figured out that if they sat down on their haunches, then they hoisted their feet and arms against the non-conducting sides, then they can escape the shock. And so they learned to beat the system, and as a result, the experiment failed. So in a couple of months, what the scientists decided to do was to use a different monkey species that didn't have as padded a posterior, and then the experiment was able to proceed. So in the end, it didn't seem like the radiation source that they used and the length and duration of the radiation caused any change in the monkey's ability to be able to flip their switches and to perform their tasks. There were other interesting experiments that came out of the animal research. This is Nancy. She was a monkey that was in the lab. She was part of a cohort of 7 monkeys that received high doses of radiation to see what the effects would be. Scientists at the time knew that if you received high doses of radiation, then you would become infertile and not be able to have healthy children. But in fact, Nancy was the only one out of this 7 monkeys to survive these high doses of radiation. And in fact, she became pregnant and was able to give birth to Chansey in 1967, who was a healthy monkey. So this surprised scientists. First of all, she wasn't expected to be fertile, and then if she did have offspring, it was expected to have mutations, but both mother and child were healthy. So this brings up a question that still interests scientists today, and that's the individual variability in response to any type of stress, whether it be radiation or pathogen infection. Why do some people are more susceptible to infection compared to other ones? In looking at the 7 monkeys in this particular cohort, it seemed that Nancy had a super ability to maintain her bone marrow. So that might be a reason why she was able to generate new blood cells, even though she was irradiated with this radiation that killed the other monkeys in the cohort. And if we had the molecular tools available back then that we have today, we may be able to answer more of these questions using molecular biology of looking at genes and DNA and what genes might be involved in this resistance. And some of this is becoming interesting, again, to a lot of these funding agencies that we have these days. But in this particular case, we don't have samples from Nancy anymore, but it's a good illustration of this variability in response to stress. We also had a series of mice experiments. So of note was a 12-year study involving the irradiation of 45 generations of mice. So this would translate to over 1,000 years of human life and generations. The experiment was to irradiate male mice and then breed them to see whether there was any long-term effects on the progeny. The mice received 1/3 of a lethal dose, so pretty high dosage. But after 12 years, it was found that even though the ionizing radiation in large enough doses will cause mutations, these mutations are generally lethal, and the resulting progeny are still remain healthy. But this is one of these long-term studies that you're able to do with a small mammalian model system. Los Alamos is also famous for the developments of flow cytometry, the first cell sorter. So this was developed by Mac Fulwiler in the mid-1960s. He combined two technologies at the time, this automated cell size analysis called a culture counter, which allows single cells to be focused into a single line. And then he combines it with this inkjet printing technology. So then the cells are put into a conducting fluid, and you're able to put charges on these cells and then separate out these cells by charge. And there have been a lot of developments to flow cytometry over the years. One major one is using fluorescent dots in order to separate out these cells. And these days, there have been lots of uses, applications of flow cytometry to separate complex mixtures of cells to understand mechanisms of cancer and other diseases. So probably all major universities and hospitals use flow cytometry in some way. The lab here has 4 or 5 different patents revolving around flow cytometry. The most recent one is this Attune acoustic focusing cytometer, which uses ultrasonic waves in order to focus these cells. And this was only in the last couple of years. So the developments of this technology, the history of it, a lot of it has been done at Los Alamos. So we're heading into the 1980s now. A lot of advances in molecular biology had happened by this point. So if you look back into history, 1953 was when Watson and Crick determined the double helix structure of DNA. In the 1960s, the genetic code was determined how DNA encodes for proteins. In the 1970s, what was the biggest discovery was restriction enzymes. So now we're able to cut out pieces of DNA and move it into other places, other vectors, other plasmids. And so all of that prior knowledge has set up molecular biology for a lot of discovery, especially in relation to unraveling the human genome. And Los Alamos was poised to make a lot of these contributions. Los Alamos is well known for its advanced computation and data analysis. So we contributed, T division, the theoretical division, contributed to the development of GenBank. In 1979, there was a concerted effort to look at DNA sequence, and folks here started the Los Alamos sequence database, which turns into GenBank. So this is a database of all known sequences from 100,000 organisms. So probably there's about there's on the order of 150, 200 billion nucleotides in GenBank. as of this year, detailing all the different genes that are known across all these different organisms. And most researchers today use this type of capability in order to look for commonalities between genes, between different organisms, and to look for interesting genes that they might want to study in more depth. Another project that we were involved with, it was a collaboration with Livermore, was this gene library project. So with the flow cytometry that I mentioned earlier, we had gotten very good at separating out cells. But the next step that scientists here took was to separate out all the 23 human chromosomes. So what they were able to do was to separate out these chromosomes, chop up the DNA, and then put the DNA into some plasmid vector so that it would be stable. And for many years, what we did was we flow sorted these human chromosomes and then made them available as a resource to any laboratory in the world who requested it. So this was a very valuable tool. Instead of looking at 23 human chromosomes for your favorite gene, you can find the gene that, find the chromosome that the gene is located on, and then just work with that chromosome. So it removed a lot of the background and furthered the study of genes more rapidly. Another venture for bioscience research is this Human Genome Project. So this started in the 1990s. The goal was to try and sequence the whole human genome. LANL, because of its earlier demonstration of capabilities in both GenBank and also this gene library project, was part of the original laboratories involved. So we were given the responsibility of mapping chromosome 16, 90.4 million base pairs, 1,300 genes, including ones that are linked to interesting diseases, obesity, autism, Crohn's disease. The genome was officially announced as being sequenced in the early 2000s by Bill Clinton. It took about 13 years at a cost of $3 billion. And as I'll mention a little bit later that there have been great advances in sequencing in the last several years that has drastically dropped the cost of sequencing and has made the potential of a $1,000 genome reality. So Los Alamos has always followed national mission, and bioscience research is no different. We've always wanted to do work that would benefit our society and our nation. So with the attacks of 9/11 in New York City and Washington, D.C., and Pennsylvania, followed closely by this Amerithrax, in which letters containing anthrax spores were sent to various media organizations, as well as 2 senators. The Amerithrax event resulted in 5 deaths and, I believe, 17 infections. Focus shifted in national mission for bioscience research. A series of legislations were passed by Congress. The U.S. Patriot Act of 2001, the Public Health, Security, and Bioterrorism Preparedness and Response Act of 2002, and then the Select Agent Program. So in terms of bio research, what these programs and legislation did was make scientists more accountable for what they do. So scientists who worked with select agents were required to be vetted by the Department of Justice. You had to go through some sort of clearance in order to be allowed to work with with these select agents. Another result was a significant increase in funding for biothreat research and preparedness. So here I pulled this graph from Science in 2005. So before 9/11, you had a small number of grants that funded this bacterial bioweapons agents type of work. So anthrax, Yersinia pestis, smallpox, those types of agents. Whereas post 9/11, you have a marked increase in the number of grants that were awarded. Whereas for other basic biology that might be looking more at environmental microbes or perhaps more clinical microbes, not necessarily bioweapons type microbes, you see a decrease post 9/11 to make up for the interest in these bioweapon agents. NIAID, which is a National Institute for Allergic and Infectious Diseases. It's the arm of NIH that funds a lot of this pathogen work. Its funding was increased by $1.5 billion in 2003. And in 2004, Congress passed the Project BioShield Act, $5.6 billion over 10 years for the purchase of new vaccines and drugs. So definitely the landscape of bioscience research changed in those 2000 years, in the year of the 2000s. So as I said earlier, I came to the lab in the year 2000. And my interests are in host pathogen biology. The pathogen I'll talk to you about today is Yersinia pestis. So Yersinia pestis is the causative agent of high mortality plague epidemics throughout history. Its transmission is by a flea vector or rodent host, and it's considered a Category A select agent by the Centers for Disease Control, the CDC. So that means that there are only a small number of agents listed as Category A, which includes hemorrhagic fevers such as Ebola, smallpox, all the dangerous ones that would be a great risk if released into the general population. Yersinia pestis is interesting. It's an extracellular pathogen. It expresses what's called this type 3 secretion system. So here's an electron micrograph of purified type 3 secretion systems. You'll notice that it looks like a syringe and needle, and that's exactly what its purpose is. It's expressed on the cell surface, and during infection, it injects virulence proteins into the host cells in order to disrupt the host and allow the Yersinia to thrive in the body. The project I will talk about, in which we look at Yersinia pestis, is this idea of the myth of junk DNA. This was a project that was funded by LDRD, which is a competitive internal research program at LANL. With the sequencing of the Human Genome Project, what we found was that only 2% of the human genome encodes for proteins. And the question is asked, well, what does the remaining 98% of the human genome encode for? And colloquially, it's been known as junk DNA because people didn't understand what the function was. And in the last several years, it's been found that junk DNA is not really junk, but that it serves an important purpose. Within this junk DNA is encoded RNAs that have a specific function to regulate gene expression. So to put that discovery into a historical context, we can go back to Francis Crick, who in the 1950s coined this term central dogma, which describes the flow of genetic information from DNA to RNA to protein. So RNA was thought for decades to be just an intermediate step that translates the nucleic acid language of DNA into the amino acid language of proteins. So it surprised scientists within the last couple of years to find that RNA has a completely unexpected function that wasn't known previously to regulate gene expression. So these small RNAs, what they do is they hybridize to the messenger RNAs, which turn into protein and prevents it from turning into protein. So this is a separate function that RNA has. And it's been termed these small RNAs, and that's what I'll refer to them as. So these small RNAs, the regulation impacts multiple national missions. So small RNAs are thought to function in plant growth and metabolism. So there are applications in bioenergy. How do you make a plant grow faster? Another place in biology where we see a lot of small RNAs is in cancer. So these small RNAs are already being found as biomarkers for cancer. Some tumors will express a different profile of these small RNAs than other tumors, depending on the different types of cancer. Also, the state of the cancer, whether it's progressed for a while compared to an early stage cancer, these cells will express a different profile of these small RNAs. So our interest is in infectious disease. Even though I gave out that number that 98% of the human genome is considered junk, in bacteria the number isn't that high because the bacteria has to conserve the use of its genome better, but there's still 20, 30, 40, depending on the pathogen, of intergenic space, so the sequences between genes, which have been called junk, and that also is found in bacteria as well. So the flow chart for the project that we were interested in, small RNA function discovery in infectious disease. We had a host pathogen system. Our pathogen was Yersinia pestis. Our host was human epithelium and macrophage cells. So what we were interested in doing was to find new small RNAs in Yersinia pestis. We wanted to see what their function was, cellular analysis to assess small RNA function and dynamics. And then we wanted to see whether we can design small molecule inhibitors of these small RNAs as potential antibiotic development. So the idea is if these small RNAs are important in virulence in some way, whether it controls expression of certain genes in the bacteria that allows it to become virulent, then if you target these small RNAs with a small molecule that blocks it from folding correctly or blocks it from targeting its messenger RNA, then you would have developed a new target for antibiotics. And in this day and age, we need as many new ideas for antibiotics as possible with the rising increase of antibacterial resistance. So for the small RNA discovery in pathogens, we use this ultra high throughput sequencing. So this is all the GATCs. When I was a graduate student, this was a very low throughput process. And here's a chart starting from 2004, where the number of bases that were sequenced, relatively small. And you can see as time goes on, it makes a big jump up because we've developed a lot of different technologies that allow for a lot of high throughput sequencing. And at the same time, this green line represents the cost per 1,000 bases sequenced. So as the technology has gotten better and better, we're able to sequence more and more, and then the cost per base of sequencing is decreasing. I mentioned before the Human Genome Project. It took 13 years at a cost of about $3 billion. The human genome was considered fully sequenced in 2003. In 2007, James Watson, who was one of the discoverers of the structure of the double helix, his personal genome was sequenced. So this was only 4 years later at a cost of about $1 million and a much shorter time than 13 years. And today, folks are talking about the $1,000 genome. There are companies out there that will do that. The cost of sequencing isn't high, but what the cost has to go towards is the analysis. So that's still holding us back, I would say. Even though we can get a list of all of these bases, what we still have to devote a lot of manpower to is determining what the significance of these bases might mean. So the $1,000 genome is definitely here, but it'll take a lot more effort to understand everything that sequence is telling us. So we used the ultra-high throughput sequencing for these small RNAs. We would grow Yersinia pestis at 2 different temperatures. So the human temperature is 37 degrees Celsius versus the flea temperature, which is 26 degrees Celsius. So this distinction, we were hoping, would be that if a small RNA is important in human infection, then it would be expressed at 37, but not at 26. So we do our RNA extraction, library generation, put it through this high throughput sequencing. And just to give you an idea of the type of data that we get, as I said before, we're looking at this junk DNA. So it's the DNA that's between known genes. So here, if we're looking at genome, we have 2 genes that are predicted or are in fact known. And what these lines here represent in black and red are different reads that we're getting in this intergenic space. So this particular part of this intergenic space is expressed at a very high level, and the red and green represents 2 different conditions. So this would be a candidate for a potential small RNA that we can further. We do a number of other molecular biology type things. Here is a northern blot, so we look at, this is an individual RNA that's recognized by a probe to our particular small RNA candidate. So we know that there's an RNA species that's recognized by our probe. We do other methods, this race sequencing, in order to get the exact sequence of this small RNA, both at the 5' and 3' ends. We can get the exact nucleotide length. The northern blot here gives us a rough estimate using these markers. It's about 300 nucleotides, but the race sequence gives us the exact sequence. So we know that it's 362 nucleotides of this particular sequence. We can do other things to look at whether the small RNA is important in infection. So overexpression, knockdown of this, what we call lanyl number 8, an sRNA sequence. So here, the control is just normal expression of this particular small RNA, but we can put it on a plasmid and express a lot of it. And here we see that there's more of this RNA compared to the control. We can add another sequence that knocks down expression. And here you see that there isn't any of this RNA. We can use these overexpressed and knocked down strains of the Yersinia in an infection assay. So if you have a host cell, you're infecting it with the pathogen. You add antibiotic to kill any pathogen that's outside. And then, so you have a host cell with whatever pathogen made it inside. You break open the cell and then plate it on plates and see whether there's as much pathogen inside in one of these overexpression or knockdown strains compared to a wild type. And what this graph shows here, in the control, where it's normal expression, you get a lot more colonies than if there was overexpression or a knockdown. So what this suggests is that there's a fine balance of what you can do. This particular small RNA seems to require a certain expression level. If you overexpress it, or if you knock it down, you don't get as much infection. So this type of biological data provides us with information on whether these small RNAs play a role in virulence or not. Other capabilities that we have at the lab are structural. So these small RNAs are about hundreds of nucleotides long. So long enough, definitely, to have a three-dimensional shape. Proteins are well known for this. There's a lot of structural information on proteins. This is transfer RNA is one of the first RNA molecules, most famous RNA molecules involved in translation. It forms this very canonical cloverleaf shape in secondary structure. But in tertiary structure, it folds in on itself, so it looks like this kind of L-shaped three-dimensional structure. So we have capabilities at the lab that can allow us to look at secondary structure and then model this type of tertiary structure. So the basis behind this is that if the RNA folds in on itself and it hydrogen bonds to other bases, then it won't be as reactive in a chemical experiment, a chemical reaction, whereas these stem loop structures are more reactive. So we do those types of experiments to get secondary structures of small RNAs. So this is just an example. We use fluorescence capillary electrophoresis. This data can be translated into figuring out the secondary structure of these small RNAs, and we've done that for a number of these small RNAs. So this is basically the first step. I've talked about the primary sequence. We use ultra-high throughput sequencing. Secondary structure, we use this chemical reactivity type of experiment to look at secondary structure. And the next steps would be tertiary structure, which we would do by our modeling capabilities. And then finally, drug interactions. So if we have a tertiary structure, we can look at how small molecules interact with this tertiary structure and perhaps prevent it from folding properly or binding to its target. And in the end, our end goal is to try and identify these small molecules as potential drug targets. And we hope to develop this type of pipeline. We're still more up in this stage, but we're hoping that we can get funding in the future to do this kind of in silico binding type of experiment. I was going to mention two other biological research types of stories that in the last year has made a lot of press. One is this HIV mosaic virus for vaccine development. So this is work by Betty Korber in theoretical division. So what she and her team has done is bioinformatically optimize these HIV virus proteins using their database of HIV diversity to try and elicit as maximal immunological coverage as possible. She collaborates with a multi-institutional research consortium in which they develop vaccines using this strategy and has tested it in monkeys. And this vaccine has been shown to decrease the probability of infection. So we've had great success in the last year showing that this therapeutic for HIV patients. And this vaccine is now planned for human clinical trials. The last thing I wanted to mention is our focus on bioenergy. So I'm sure you've noticed that there's a new building in the last year or two that's been built near the food co-op. It's this New Mexico Consortium Biology Laboratory. It's a $12 million building, 24,000 square feet laboratory, which opened in 2012. And this building is focused on developing ways to make algae grow faster, increased fatty acid production to provide more energy as competitive alternatives to crude oil. There are also efforts in plants and cyanobacteria research for bioenergy. So our bioscience division has two main groups. So one is biosecurity and public health that I'm a part of, and the other one is bioenergy focused. So even I'd say within 5 years ago, bioenergy was a very small component of bioscience research, and here 5 years later, it's whole group. So that talks about the energy focus that has become a new national mission to try and find these alternatives to fossil fuels. So that's pretty much my talk. I wanted to acknowledge folks who have helped me in preparing this. So Alan Carr is our Lanel historian, and Larry Gibbons is our Lanel image archivist that provided me with a lot of those old photographs of the old bioscience work. Rebecca McDonald is our communications specialist who made the timeline that I showed earlier. I wanted to thank many senior bioscience scientists who I talked with to get their take on the historical importance of the bioscience work. Our small RNA research team that did the work that I talked about in my lab, and also the LDRD program that funded the small RNA work. So I'd be happy to take any questions. Thank you for your time.
Questions?
Yeah, I was wondering if you could elaborate a little bit on those plutonium studies, the 18 patients and given tracer amounts.
Right.
It doesn't sound like something that can get through an IRB nowadays.
No, that was a different time.
Were they being treated for something?
Right, so they chose the patients, a lot of them were terminally ill, so they weren't expected to live long. But if you look at the patients, a number of them did live for a number of decades, even though they were predicted to not have too many years left. There's a great article about it that was printed in a Los Alamos science publication back in the 1990s, and if you're interested, you can find it on the web. It's called, if you put in human plutonium injection experiments, you'll probably pull it up, and it gives a very detailed history of how they chose the patients, the doses that they gave them, what they found out from individual cases. The work was done over a period of time, so they tried, the results that they got from like the first 2 or 3 sets of patients, they tried to learn from that before they would do the next injection experiments. But the IRB didn't exist at that time. I'm not sure if we can do something like this today. I'm pretty sure we probably can't. But they were trying to use tracer amounts, so it wasn't like a complete overdose. They were trying to see how much would be excreted and where it would go inside the body.
Can you tell us a little more about the small RNAs from your Yersinia? Are there many of them? The one that you described is kind of large for a small RNA.
Yeah.
Most of them I think are smaller.
But perhaps it's a misnomer to call them small RNAs, but in the ultra-high throughput sequencing, we probably found 20 that we would follow up on, that it had all the right hallmarks. It had a promoter and terminator sequence, which showed that it was expressed independently. It was expressed at high levels. It had a structure, if you were just put it into a kind of a structure folding program. But other studies and other organisms have found, more. And I think the hard part about small RNA research is that these small RNAs are thought to be expressed in response to specific conditions. So if you grow up a bacteria in a low nutrient media, it'll express a different set of small RNAs compared to the bacteria in a high nutrient medium, because the small RNAs are being expressed in response to its environment. So in a way, I think when we do experiments, you can only grow it up in one type, and so you get your set of small RNAs. But there's probably a vast underrepresentation of the small RNAs that actually can be expressed by an organism, because there are so many different conditions that you can possibly grow up a bacteria in. So there are probably thousands across all different organisms now. They're called by many different names, depending on their length, depending on their function. So there's a whole field of RNA biology that's cropped up in the last 10 years, I would say, with these different functions from RNAs, which wasn't appreciated before.
Are these made by the same polymerase that makes messenger? And are they capped?
I don't think they have that same. So I don't think they're like messenger RNAs, to my knowledge.
So these are made in the bug itself, not by the host?
Yes, they're made in the bug. There are small RNAs in the host as well. There are really small ones on the order of 20 to 25 nucleotides called microRNAs. And those are the ones I was referring to when I was talking about cancer biomarkers, are microRNAs. So there are different categories. The small RNAs that I'm talking about in bacteria are more like several hundred nucleotides long, and they're different than these other microRNAs.
So these are made all
by the bug.
Polymerase bug.
Yes.
Can you just ask it? Can you just use, I'm
Okay.
afraid of feedback. If you could repeat the question.
Okay. Your description or summary of some of the earlier work on the effects of long-term exposure with regard to healthy progeny reminded me that a lot of research has been done on the similar effects or suspected effects after the Hiroshima and Nagasaki exposures.
Mm.
And it found very similar. It sounds very similar to what you found. That made me wonder, has that research impacted or contributed or related in any way that's important to yours?
To my current research, you mean?
Yeah.
Perhaps not directly. The mice experiments that I spoke about, and also the Nagasaki events, were high dose exposures. So you would expect that there would be a lot of initial death, but not as much effects later on. But another area of research which followed that was low-dose radiation. So instead of having whatever a high dose where the effects would be lethal, you're looking at very small doses. And with the Fukushima events of just a year or two ago, where the nuclear reactor in Japan broke apart in the tsunami, a lot of that, there's a lot of interest again, I think, in radiation effects. Even though we deal mostly with biothreats of pathogens, there's still this low-dose radiation effect in case there was some sort of dirty bomb or some risk of that sort in today's world. So I would say low dose is interesting from our point of view, because no matter how you stress out a particular host, some of the effects will be the same. It'll be like an immune response, some sort of inflammatory response. There'll be differences, but some of the core groups of like survival type cells or stress type of cell responses will probably be the same. So I can see some overlap there where we have low dose radiation and compare that to pathogen infection.
My son-in-law is in Navy, and he was reporting about the aircraft carrier that was off the coast of Japan shortly after the tsunami or the explosion of the reactors over there. And they were pulling in highly radioactive water through their desalination equipment, unknowing to them. And there have been a series of tumors and cancers and leukemias. Have you heard anything about that?
Sorry, I haven't myself. I think probably there will be literature reports on that as the epidemiology of that event occurs, and they'll be able to look at it more at the long-term effects of that, whether there are along cancers that have developed over time. But I don't know of that work myself.
Well, if you can move about 3 feet that way, it'll fill that center.
Okay.
Now you're talking.
Thank you.
A lot of the information you're developing would seem to have a lot of interest to commercial drug companies. What, if any, relationships do you have with commercial drug companies, either intellectual or financial?
Yeah, I personally don't with the work that I'm doing currently. We do a lot of fundamental basic biology, which is of interest to companies, I think. But a lot of companies, they're more interested in probably more mature types of possibilities. So for example, with this small RNAs, this number 8 that we're looking at, that we've gotten the furthest along in, if we're able to get a three-dimensional structure that we can show by some sort of NMR or X-ray crystallography or something, and then we can, if we're able to screen a library of small molecules and identify 5 or 10 that may prevent it from folding correctly, at that point, I'm pretty sure we can get a drug company to be interested, because we've done a lot of the upfront work, and they have to do all the good the lab practices and the drug safety and the cytotoxicity type experiments that we don't do here at the lab. So it's a partnership, and the lab has a technology transfer division that looks at intellectual property, and there are a number of bioscientists who've are plugged in to that area for development of potential antibacterial drugs and the like. But at least for the work that I do, we haven't gotten that far yet.
Any kids want to ask their mom a question? Or a husband, his wife? Are you sure? All right. Any other questions?
I know Sandia Lab has done a lot to assist the government to prevent terrorism attacks, say in airports or public places. Is the lab involved in those programs separately or together with Sandia?
Do you refer to bioterrorist attacks or anything?
I guess I do. I don't. This is a whole new world for me.
Yeah. So I would say we don't have anything practical, currently. But things that we do, there's a BioWatch program in which it's currently operational. There are various filters on top of holders in major cities around the country, and the filters are collected every so often and to see whether there are dangerous pathogens in the air. It's a biosurveillance type system. So we have a program that tries to develop some of these assays to see whether we can rapidly detect pathogens. A lot of the work that we do, a lot of the biosecurity work that we do is geared towards developing a better way to detect a pathogen, a quicker way, a gene sequence that defines it as it can only be found in one pathogen and no other pathogen. So it's still a lot of basic biology. Looking for these biomarkers is usually what we call it, a biomarker of host infection, a biomarker of the existence of a pathogen. So that type of work we're involved with.
have one. I got a Gen 2.0 for Christmas. This is where you send your a swab with your saliva into
for your
National
DNA
Geographic.
sequencing.
But not the whole sequence. I think it's just it sort of traces.
Markers.
I haven't read it. Is it safe to do that?
Yeah, I think so. I think it's always what you do with the information. I think you have the information and you can find that you're possibly going to develop Alzheimer's in another 30 years, and maybe that will change your behavior somewhat, but maybe not. So.
All right. Any final questions? All right. Let's give Dr. Honggeller a big round of applause.